The overall goal of this experiment is to characterize plant cell wall polysaccharides.
This method can help us answer key questions in the plant cell wall biology field, including glucomannan structure and quantity. The main advantage of this technique is that it doesn't require specialized training or expensive equipment.
Generally, individuals new to this method will struggle with interpretation of the gels, so inclusion and understanding of all the controls described is critical.
The alcohol-insoluble residue, or air, was previously prepared from harvested plant material as described in the text protocol. Weigh 6 milligrams of air into a 15 milliliter centrifuge tube and add water to a final concentration of 2 milligrams per milliliter. Vortex to achieve an even suspension. Aliquot 500 micrograms into microfuge tubes. Dry aliquots using a vacuum centrifuge at 30 degrees Celsius overnight. Pre-treat the air, including an aliquot for a no-enzyme air control, with 1 molar sodium hydroxide for 1 hour at room temperature. After 1 hour, add 200 microliters of water and 20 microliters of 1 molar hydrochloric acid to each tube to neutralize the sodium hydroxide. Test that the pH is about 6 to 7 by removing 1 microliter and spotting it onto paper pH indicator strips. Add 50 microliters of 1 molar ammonium acetate buffer pH 6.0.